This is a review of reproductive system. It'll be the last video that I make for AMP2. And this information obviously goes along with the kidney information covered in a different video for the final exam. So the first thing I want to do before we branch off into separate male and female reproductive physiology is a very quick bit of recap about mitosis and meiosis as it relates to reproduction. So we're pretty much going to leave mitosis alone, except to say that the entire purpose of mitosis is for cells to completely copy and clone themselves. So if you start with a cell, as all adult human cells are, that are diploid. Diploid, or 2N, meaning they have two copies of every allele, or I should say two copies of every gene, maybe two different alleles, two copies of every chromosome. So two chromosome 1s, two chromosome 2s, two 3s, etc. And then, of course, either two X's or an X and a Y. In mitosis, the purpose is make an exact copy of all those chromosomes. So you start with 23 pair, right? Through mitosis, you double, so you end up having 4 sets of each chromosome. But then when you divide into two cells, you're back to 23 pair again. So the two daughter cells that come out at the bottom of mitosis should be, as long as there were no errors, should be exactly the same as the cell that you started with. Meiosis starts off as a very similar process. You make copies and double all of your chromosomes. The only difference is, well, actually there are more differences. The big one that we're going to talk about here is, instead of just dividing once and splitting into 2 cells, you divide twice and split into 4 cells. Now the end effect of that is, instead of ending diploid, 2 copies of every chromosome, 2 copies of every gene, like we did in mitosis, you're ending haploid. 1 copy of every chromosome, only 1 copy of each gene. And the main point to take away is the reason this is necessary is, and the whole goal of the reproductive standpoint, or reproductive system from a biological standpoint anyway, is simply to get haploid cells from a male connected to haploid cells from a female. And so if both male and female contribute a haploid cell that each have 1 copy of each chromosome, when egg or oocyte and sperm meet up, the new embryo that results from that is going to be back to diploid. And so all cell division outside of reproductive tissue in a person is mitosis. Only in reproductive tissue do you have meiosis, because it's the only place where it makes sense to cut your number of chromosomes in half. The other difference that occurs is something called crossing over, which we may or may not have time to mention in class, and you've probably seen in a past class, but it won't really be part of the exam for this section. Okay, for male reproductive, we'll mention the anatomy that's necessary as we go, but I'm not running through the whole anatomy list since we have plenty of time to do that in class and you have your lab list. So there are a few topics, really not that many to cover in male reproductive, that are important, but it's fairly simple. The first is sperm production. And what's essentially happening here is inside one testis you have a very large number of these coiled tubes, okay? And we call those seminiferous tubules. And the wall, the epithelial wall of those tubes, is full of cells that are constantly going through division. And they're actually going through 2 different kinds of division, mitosis and meiosis. And the reason for that is, if you look at the wall of that tube, which you can see here, and which is maybe even more clear on the next slide, the inside of the tube is filled with developing sperm cells at the center of the lumen. Those are very close to becoming mature sperm, or spermatozoa, if you're going to use a zoological term. These immature cells around the outside, which we call spermatids, since these sperm are always moving through the tubules, to leave the body eventually, the entire process has to involve these cells migrating in this direction. Now the problem is, if all these cells did was go through meiosis, meiosis ends. With mitosis you can just keep dividing and dividing and dividing over and over again. But with meiosis, when you hit the end, when you get to those final 4 haploid cells, it's over. Those 4 cells are the end product, and you can't keep going. And so what ends up happening is this very outer ring of cells right here, these cells are going through mitosis and dividing. And very similar to the cells at the base of the epidermis, very similar to the cells in the esophagus, hair follicle that turn over a lot, these are stem cells, and they keep dividing over and over again. And when one cell splits into 2, and imagine this cell divided and split into these 2 cells, one of them will stay here and continue dividing as a stem cell, and the other will start moving this way and migrating toward the center, toward the lumen of the seminiferous tubule. And the cells that migrate and move inward here will start going through meiosis. And so you need mitosis around the edge to keep dividing and turning into new cells so you don't run out of cells. And you need meiosis toward closer to the lumen because they're going to become mature sperm cells. They have to go through meiosis to get from diploid down to haploid, right? Because each of these sperm cells should only be carrying half the genetic material, half the chromosomes that man has. Okay, so this process is happening in all of these coiled seminiferous tubules, and they all connect back to another duct that they all merge into called the epididymis, where they continue maturing, developing into, complete mature sperm cells. Once that happens, they'll leave the area around the testis into the ductus deferens or vas deferens, as you've probably heard it called. Okay, and from there, you're moving out of the body potentially, which we can get to in a little bit when we talk about a bit more anatomy. The other thing that you can see on this section is surrounding the seminiferous tubule is another small population of cells called interstitial cells, or sometimes Leydig cells, after the person that first described them. Those are your testosterone-producing cells. And these are the 2 big jobs or roles of the testis. One is make sperm, the other is produce testosterone. Okay, so while those cells are going through this process, they're changing their shape. We call this morphology. The cells are going through morphogenesis. And they have to convert themselves from these normal, round-looking, nondescript cells to a very unique, very polarized cell. And if you look at a sperm cell, you'll see a couple things. The thing that most people have seen and are aware of is the tail, which is actually a flagellum that flips back and forth to actually propel sperm through fluid so they can move. Just above that, this mid-piece right here, which gets slightly larger, is mostly filled with mitochondria to produce ATP that are used to run the flagellum, to make it move. Above that, you'll see a nucleus. And remember, that's the entire purpose of a sperm. All you're trying to do is deliver that haploid nucleus to the oocyte. And so the rest of the sperm is really just a carrier or a vehicle for the nucleus. The last thing you see at the very tip is a tiny little pocket of digestive enzymes called the acrosome. And what this does is, when the sperm finds the oocyte and connects to it, it's going to break apart that acrosome, release the digestive enzymes, and that's what's used to actually burrow a hole through the oocyte to get the sperm nucleus inside. At that point, the nuclei can merge, and you're on your way to having a new embryo. All right, so jumping back where we last left them, these maturing sperm, remember, they were made in the seminiferous tubule. They matured moving through the epididymis, and they're now stuck in the ductus deferens. And from there, the ductus deferens is a fairly long tube, moves back up and wraps around the bladder, coming down by the seminal gland, which produces the bulk of the fluid or semen that goes along with sperm to get out of the body, makes its way through the prostate. Prostate adds some stimulants that start getting sperm moving so they begin swimming. Because the whole way through this ductus deferens, sperm aren't actually swimming. They're being propelled by smooth muscle tissue, which I'll show you a little bit later. And you don't want them to actually start burning through their ATP and start swimming until they're about to leave the body, because the purpose of the flagellum is not to swim to get them out of the male. The purpose of the flagellum is to start swimming to seek out the oocyte once you're inside the female. Okay, so in order to make that happen, there are certain mechanical considerations that need to be had. So, for obvious reasons, you can't very easily get sperm out of the flaccid penis, and so we've got to have an erectile response. And that's mostly produced by, almost entirely really produced by changes in blood pressure. So the way this system works is, if you are unlucky enough to have your penis cut in cross-section, this is what it would look like. And there are two large open spaces on the superior side that are called corpora cavernosa, and one smaller open space at the bottom, the corpus spongiosum. And all these really are cavities that can be filled with blood and can tolerate pretty high pressures. So the response, which is entirely stimulated by the nervous system. So after arousal, you'll expect nerves from the brain to stimulate the production of nitric oxide in the tissue surrounding the arteries leading into the genitals. And remember, nitric oxide is a vasodilator, and so that's going to open up those blood vessels. It's going to dramatically increase blood flow, which will increase blood pressure. The way the anatomy is set up, the increase in blood flow that makes the tissue swell, right, this corpus spongiosum mostly, also pinches off the veins carrying blood away from the tissue. And so you get a huge amount of blood flow in, increasing pressure, but relatively little blood flow out. This is unique in that you wouldn't want it to happen in most organs because that cuts off flow. But if you think about it here, the purpose of the increased blood flow is not to supply the tissue with oxygen and nutrients and things like that. The blood pressure is being used as sort of as a hydraulic pump of sorts. All you're trying to do is inflate the thing. You're not really trying to get it more nutrients. So a lot of blood flow isn't important. Okay. Of course, after arousal goes away or after ejaculation, the nitric oxide levels will drop again. And this is, the nitric oxide thing is perhaps best known because enzymes that degrade nitric oxide are the primary target of erectile dysfunction meds like Viagra and Cialis, things like that, which were originally designed, or the hope was that they would be designed to combat hypertension by relaxing systemic arterioles, but they happened just by chance to stumble across an inhibitor that specifically blocked nitric oxide degraders in genital tissue, but not in others. Okay, so at this point, we've made the sperm. They're sitting in the ductus deferens. We've gone through, this whole circus to prop the thing open. Right? The corpus spongiosum, by the way, doesn't contribute a whole lot to the actual erection, but does keep the urethra open to make sure that sperm and semen can get through. And this is where the histology of the ductus deferens makes sense. Again, remember, sperm are not swimming out of a person. They're being ejected by force. And so if you look at the ductus deferens, you see a relatively small lumen in the center, a little bit of epithelial tissue around it. Everything else, all of this, is smooth muscle. Okay? The entire purpose of this is to propel sperm forward. So very much like the peristalsis of the smooth muscle in your digestive system keeps moving foodstuffs and chyme through the digestive tract, the smooth muscle of the ductus deferens is what propels sperm, and with it later on semen out as an ejaculation. Okay. So at that point we're pretty much done, and we can pick up, everything's really in the hands of the female reproductive system at that point. And so the last thing I'll mention is a little note on sperm production and sperm levels. And what I'd like you to know for the test is, Sperm production in men is very important, and it takes different roles at different times. And so if you look at the course of, this is very smoothed over, very normalized sperm levels over the course of one man's lifetime. And you'll see a few things. If this is birth right here, right, there's birth, this is gestation. The first big surge in testosterone actually occurs in utero around 3 to 6 months gestation. And the purpose there is, this is what's giving you what we'll call primary sex characteristics. That's mostly just reproductive organs. So this is just developing male anatomy that otherwise would have sort of defaulted to female anatomy if the Y chromosome wouldn't have been there to direct that big surge in testosterone. The big surge at puberty that everyone's familiar with produces secondary sex characteristics. That's producing everything other than basic anatomy that you associate with adult males versus adult females. So this is body size, muscle and bone mass, facial hair, deeper voice, everything that you associate with a male, post versus pre-puberty. The other one that you'll see is a little blip right after birth, in the first year of life. There's not a huge amount of data here, but it is known that differences, pretty strong statistical differences in male and female brain anatomy and function can already be observed at that age, at about 6 months old. And so the best hypothesis that anyone has going right now is that small spike in testosterone after birth may be what begins to masculinize the male brain and distinguish it from female brain. And the last thing, which I touch on a little bit, is testosterone replacement therapies have become very common, especially for middle-aged and older men. The idea being that, if you look on a population basis, they have it here, testosterone levels trailing off at age 70 or so for this man, but in reality, testosterone levels on average start to drop as early as the late 20s for a lot of men. And it's easy to brush this off as being, okay, well, testosterone's not that big of a deal. If you don't have a lot, you just maybe not quite as, strong and aggressive and things that we don't care too much about anyway. But it really does have a big effect on a lot of things. Depressed testosterone level is strongly associated with obesity and heart disease and general, lethargy and tiredness, decreased strength and physical ability. And so this is really important, really for women too, but there's not nearly as much data on female testosterone levels. And anyway, what a research group from Australia showed is that if you sample men who meet a certain criteria, being they maintain a healthy body weight, they get plenty of sleep, they don't smoke, they don't binge drink, and generally take care of their health, testosterone levels are really flat. They don't have to decline. And the conclusion from that, what seems like the most obvious conclusion, is that the decline in testosterone is probably mostly caused by people taking care of themselves less, or at least the accumulation of poor behaviors over time. It's pretty hard to suppress testosterone levels in an 18-year-old, but after growth and puberty are completely over and a man's in his 30s, 40s, 50s, things can start to go downhill and have negative effects. Okay, so at this point, I'm going to move on to female reproductive physiology, and we're going to start off in the ovary, and then we'll move on to the uterus, and then we'll sort of take that through to pregnancy and childbirth, at least briefly. So the most important thing that's happening in the ovary is, well, a couple things. Some estrogens produced here, that's very important. But the big physiological event that's happening is the development of a follicle. And just a bit on terminology. When you see, I'm going to jump ahead here for a minute. When you see a whole ovary in cross-section like this, all of these things that I'm showing you here are follicles. Even these little tiny things up here are what we call primordial follicles. And every menstrual cycle, one follicle is sort of released or moves from the primordial follicle pool and will start moving this way over time, sort of clockwise around this picture. And they'll eventually get a little bit larger, Right? These larger ones we'll call primary follicles. They'll get even larger yet down here, secondary follicles. And then, most commonly one at a time from the ovary, they're released. Okay? That release, which is sort of shown down here looking very volcanic and fiery, is that is ovulation. Right? The oocyte ruptures out of the follicle. Okay? And those two terms, oocyte and follicle, we need to distinguish. So if you zoom in very close on a secondary follicle, this entire structure, right, this whole thing, is one secondary follicle. Right? This enormous cell, it really is very, very large. At the center of the follicle, that's the oocyte. Okay, so the oocyte is the very, very large cell at the middle of the follicle. It's the cell that's going through meiosis. It's the cell that's going to actually receive the sperm to become the new embryo. And so the rest of this surrounding tissue is part of the follicle, but it's not actual reproductive cells. The corona radiata refers to this, multiple layers of supporting cells around the oocyte. Gets its name because it sort of looks like a crown radiating around the oocyte, apparently. And this fluid-filled space that's packed full of fluid reserves and nutrients, that's the intrum. Okay? And so the thing that's happening inside the ovary is the follicle's growing, the supporting cells around the oocyte are dividing to increase in number, and the oocyte itself is getting larger, and eventually it's going to start moving through meiosis. And the whole goal is to prepare that follicle so that when the timing is right and you're ready for ovulation, the oocyte can burst out of the follicle, enter the uterine tube or fallopian tube, and be ready for fertilization. And that's what's happening here. Okay, so remember the time scale of this as I jump around. All follicles start off as primordial follicles out here. One at a time, they're released and move around the cycle. Now remember, it doesn't take one cycle or 1 month to get from here to here. It takes 1 month to move to this step, another month to move to this step, another month to move to this step. Right? And then once it's finally matured and ready, then the follicle's released when it gets to the last position on this movement. The other way that we can look at this is on sort of a timeline scale of events in follicle development and meiosis. So the very top of this pathway is showing you how you make primordial follicles, and this happens entirely before birth of the mother in this case. So before the female is even born, during gestation, a cluster of cells goes through a lot of mitosis, right? Not meiosis yet, but just meiosis to build up a big population of primordial follicles. And those tiny primordial follicles are just going to sit there and not really do a whole lot of anything all the way through infancy, childhood, all the way up until puberty. And as soon as puberty hits and menstrual cycle starts occurring, right, those primordial follicles are going to start moving through the ovary and start growing into primary and secondary follicles that will eventually be ovulated. And at that point, once they start maturing in the follicle, they'll also start going through meiosis. Now, I'm not worried about prophase and metaphase and all the different stages. I just want you to know that it starts happening. And of course, the whole point is you need to get down to being a haploid cell. The other thing to consider, though, is meiosis ends with 4 haploid cells, and we only want 1. At least most people only want 1 baby. So you end with 4 cells, but you only need 1. And so all the nutrients and all the materials are packed into one large oocyte or ovum. The other 3 haploid nuclei that would have formed the other 3 cells, those get thrown away as polar bodies. They essentially just disintegrate and go away because we don't need 4, we only need 1. Okay. And maybe last point here, it should be noted that last polar body leaving, that signifies the absolute end of meiosis, meiosis 2. That only happens if sperm penetrates. So if sperm gets inside the oocyte, meiosis II completely finishes. You've got an embryo at that point once the nuclei fuse, and everything's over. If the oocyte never gets fertilized by sperm, you never actually completely finish meiosis. All right, so that's getting the follicle and the oocyte ready in the ovary. But once the follicle's ruptured and you've ovulated the oocyte, the other thing that has to be happening is, in case there is fertilization, or if there's going to be pregnancy, the uterus has to be ready for that. And this histology is showing you uterine tissue, and this entire layer right here is the endometrium. It's filled with these endometrial glands that secrete all sorts of important products into the interior of the uterus, and the epithelium right on the surface. That epithelium and the endometrium below it, that's where the embryo is going to implant if there's fertilization. Okay. And what we have to talk about is the hormonal regulation of how endometrial growth is controlled. Okay. So this is the tissue that grows and then degrades and then grows again during menstrual cycle. And the thing that has to happen is the endometrium needs to be at its fullest and thickest right when implantation would happen. And then if there is no pregnancy and there's no implantation, the tissue will essentially fall apart, degrade itself. Right? That's where you see the loss of blood at the end of the menstrual cycle, and then the whole cycle will start over again. And so this set of graphs is showing you how these things are coordinated. I say coordinated because this top portion is showing you when ovulation occurs, average day 14, but it can vary a little bit during cycle. This graph is showing you endometrial growth, smaller to thicker. Okay. The reason this has to be coordinated is if ovulation happens at day 14, implantation is likely to happen 3 to 5 days after that. And what you'll notice is that window down here is when the endometrium is at its thickest. Okay. So you have to have that endometrium large and vascularized so it's ready for implantation at the right time. If the endometrial growth peaked back here before ovulation, that wouldn't make any sense. You'd be out of sync. Okay. The hormone that keeps all of this in sync is estrogen. Okay. And estrogen does two things. Right. Estrogen produced by your ovary stimulates growth of the endometrium, and so you see this endometrial growth rising the same time that estrogen levels are rising. And at the same time, when estrogen levels peak, it also stimulates the release of LH and FSH, luteinizing hormone and follicle-stimulating hormone from the pituitary, which we talked about back in the endocrine system. Okay. LH and FSH are the triggers that cause ovulation. And so what you have is a coordinated response here. At the same time that estrogen is making the endometrium grow and thicken, it's also triggering ovulation at just the right time. That keeps things in sync so that ovulation happens just before the endometrium reaches its peak growth, because that's when you would like implantation to happen. Okay. Now, it may seem strange that, why would the ovary produce estrogen to tell the anterior pituitary to release LH and FSH, which just goes right back to the ovary to trigger ovulation? The most reasonable assumption here is that the point is to keep your brain in the loop. Right? Since the anterior pituitary is connected to the hypothalamus, this is really discussing things with the hypothalamus, letting the brain know what's going on with the ovulation cycle. Partly because female behavior can change quite a bit depending on time of cycle. Okay, so ovulations happened at that point, and the rest of this graph is assuming that there's no fertilization, that there's no pregnancy. If there's no pregnancy, what happens is this tissue that's left over, right after the oocyte is ovulated, there's some tissue from the follicle that's left behind in the ovary. This becomes something we call the corpus luteum. And the corpus luteum produces another hormone called progesterone. You see that in the green line here. And so as soon as ovulation happens, progesterone levels rise because the corpus luteum sort of comes into existence. And you need progesterone to keep the endometrium growing and thickened. If there's no pregnancy, the corpus luteum will degenerate, fall apart, and go away, which means that it'll stop producing progesterone, and progesterone levels fall. If progesterone levels go down, that's what causes the endometrium to fall apart, and that's what you see as the blood loss during menstruation. If there was a pregnancy, things would be very different, and we'll talk about that in just a minute. Okay. Right, so assume there is pregnancy. Sperm, attacking the oocyte, it seems, will dock with the oocyte membrane. They have receptors on it that will lock the sperm into place. At that point, the sperm will release its acrosome. Right? Remember those digestive enzymes kept at the tip of the sperm? It will burrow its way in and drop off a nucleus. The goal is to have that sperm haploid nucleus merge with the haploid nucleus of the oocyte, and also to make sure that more than one sperm don't get in. And we didn't talk about the details of it, but the oocyte will actually go through a distinct chemical reaction in order to block multiple sperm from getting in the oocyte. That would give you too many chromosomes. You can't survive like that. That would lead to a miscarriage if it happened, which does occasionally happen. Okay, so we've got the sperm in. The oocyte's fertilized. This is happening somewhere in the uterine tube between the ovary and the uterus. This also shows you why the oocyte had to be so large. The primary reason is this oocyte has to get from the top of the uterine tube all the way down into the uterus, where it can implant into the wall of the endometrium right here. That takes several days, ballpark, half a week to closer to a full week. That means that this oocyte has to survive nearly a week without blood flow, because while it's floating down the uterine tube and just beginning to implant, there's no blood supply. And so these cells, they start going through cell division, and since the oocyte and now the new embryo is so large, it can divide, but the cells don't really grow. And so it divides very rapidly. The cells keep shrinking, going from large to smaller, 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 as cell division occurs. After the first week, once the embryo is securely implanted and has some blood flow, then it can start getting new nutrients and really growing. Okay, so you need that large cell size to hold the oocyte over, the embryo over, really, until blood flow is established. Okay. Once fertilization happens, the other thing that we need to talk about is we need to make sure that this doesn't happen. Okay? This graph is showing you what happens if there's no pregnancy. If there is a pregnancy, the endometrium can't fall apart and degrade, but you'd lose the tissue that the embryo is supposed to implant into. And so we have to do something to prevent those progesterone levels from falling. We want them to stay up, right? So the endometrium continues growing and doesn't degenerate. That something is a hormone called HCG, human chorionic gonadotropin. And HCG is the perfect signal because it's produced by the embryo, not by the mother. And so as soon as fertilization occurs, the new embryo will produce HCG. HCG, its target cells are in the corpus luteum. That's the growth stimulus to keep the corpus luteum around and have it continue making more and more progesterone. That's why progesterone levels rise. That's why estrogen levels rise. And this is what continues growth of the uterus, development of the placenta and the umbilical cord and everything. So HCG is really the trigger that separates a menstrual cycle with no pregnancy from one with pregnancy. HCG also happens to be the easiest way to test for pregnancy. And so both home pregnancy test kits and lab tests, at least early on in pregnancy, they're just testing for the presence of HCG. Now, after HCG rises and falls, estrogen and progesterone are going to keep increasing. And estrogen especially, which is being produced by the placenta at this point, is estrogen sort of the timer that sets the stage for when birth is going to happen. And the way that it does that is, as estrogen levels rise, at some point they get high enough that they stimulate the activation of oxytocin receptors in the uterus. And oxytocin is the hormone, remember, coming from the posterior pituitary. And the thing that it does, in this case at least, is it stimulates uterine contraction. And so once estrogen levels get high enough, you activate oxytocin receptors, and from that point, this initiates a positive feedback loop, right? One of the very few positive feedback loops that we talk about. And it's positive in that oxytocin stimulates two things: contraction and the release of another set of hormones called prostaglandins. Prostaglandins stimulate even more contractions, but they also stimulate more oxytocin. Right? So everything in this loop is positive. There's no negative to break the cycle. And so more oxytocin, more contractions, more prostaglandins, more oxytocin, more contractions. It just keeps running around in a circle until contractions get intense enough and spaced close enough together that you can get the baby out. Okay, backing up just a little bit, we should take a closer look at placental blood flow. The thing that I really want you to take away from this is to remember that the mother shares nutrients and removes waste products from the blood of the fetus, but their blood supplies are completely separate. They don't mix. So you have maternal blood vessels here, and you have fetal blood vessels coming off the umbilical cord here, and they sit right next to each other, next to these big branching structures called chorionic villi. There's branched capillaries. And so the two blood supplies come in very, very close contact with each other, but they can't mix. And the most important reason for that is you don't want whole cells mixing, right? Red blood cells, white blood cells. Some of the blood components of the fetus could potentially be seen as foreign antigens to the mother, and so whole cells should never mix. Smaller things, obviously nutrients, waste products, even fairly large proteins like antibodies can pass through. That stuff's fine. Whole cells should not pass back and forth between maternal and fetal blood supply. All right, the last thing that we'll talk about here is milk production. And I'm not going to go through a lot of detail except to say, remember that mammary glands are exocrine. So this is, they're actually producing milk by breaking off fragments of the cell itself that are being released into the product through those glands. And the ducts are surrounded by smooth muscle. So you're producing the fluid and then using smooth muscle to push it out at the right time. And the right time is something that's initiated mostly by mechanoreceptors. And so mechanoreceptors sense the movement produced by the infant trying to feed. And that initiates another positive feedback loop. Okay, we haven't seen many, but two of them are right here. And so the thing that'll happen is that mechanoreceptor activity will stimulate the release of PRF, prolactin releasing factor, in the hypothalamus, which stimulates the release of prolactin then. The name makes sense. Prolactin for stimulating or encouraging lactation. Okay. This will increase milk production, and at the same time, it's also going to stimulate oxytocin. And just like oxytocin stimulated uterine contractions during birth, now it's playing a slightly different role, but it's still stimulating contraction. Instead of stimulating the muscle around the uterus, it's stimulating the muscle tissue around the ducts of the mammary gland. This is what's called the letdown reflex. So you move milk through those ducts, it's pushed out, and the positive feedback loop comes from the fact that the infant is part of this loop. So the more milk you let out, the more the baby likes it, the more they try to feed, the more you stimulate the response. And it works as positive feedback because there's a distinct endpoint. Right? So just like childbirth, you can use positive feedback because once the baby's born, it's all over and the system resets. With milk production, the endpoint is eventually you run out of milk. And a child can feed much faster than milk can be produced. And so once the milk's all gone, the baby will stop getting that feedback, it'll stop getting fed, we'll stop suckling, and the whole thing will shut off.